Hello, welcome to Science Hangout. I'm Sadashiva Pai. Today we have Dr. Alysson Muotri, Associate Professor, University of California, San Diego School of Medicine, Department of Pediatrics and Cellular and Molecular Medicine is with us. Dr. Muotri has won several distinctions and awards to his credit. And I'm going to mention only a few of them here. In 2009, he won National Center of Leadership in Academic Medicine, UCSD. In 2009, again, he won NIH Director's New Innovation Award. In 2000, Emerald Foundation Young Investigator Award. In 2010, he was 100 most influential people in Brazil. In 2000, Stigma Buster from National Alliance and Mental Illness Award. So I can go on, and I'll mention a couple of them at the end. In 2016, he won Faculty Innovator Award from UCSD, and this year already he has won a Leadership in Research Award of Excellence by Children's Hospital. In addition, he is in the review board of several publications, and he has won several of grants and all. So welcome, Dr. Alysson, to Science Hangout.
All right. Thank you very much. Thank you for the invitation, and thank you for organizing this. I mean, the more we spread about the science, the better, I feel.
Okay, thank you very much. Yeah, let's get going. Let's talk about, can you please tell me about yourself, a little bit about your background, education, etc.?
Yeah, so I'm actually originally from Brazil. I got my PhD in genetics in 2001. And in Brazil, we don't have like a very strong neuroscience community. I always wanted to get involved with neuroscience. So I felt that I was, after the PhD, was the time to go abroad and explore a little bit as a postdoc. So that's when I joined Rusty's Gage lab at the Salk Institute, work with neurostem cells, neurogenesis. And I was a postdoc there for several years. We have very nice publications. Were a very productive time for me. After that was in end of 2008, 2009. I became a assistant professor here at UC San Diego and established my own lab. In the lab, we are mostly focused on neurological disorders, try to model using human stem cells. I was very fortunate because during the postdoc period, I was able to learn how to grow human embryonic stem cells. And when I start my lab, that was when the whole IPS revolution, induced pluripotent stem cells revolution from Shinya Yamanaka, appear in the literature. So I was able to jump into that technology and was among probably the first users of the IPS reprogramming technology to model neurological disorders. So since then, my lab has been focusing on comparing disease models with controls and try to understand not only what happens when you have a specific genetic alteration, what happens to your brain cells, to your neurons, to your astrocytes, but also use that to screen potential drugs that may be able to ameliorate or even revert the neurological disorder that we are studying. So I feel that this is a very exciting model because it's not a static model. And let me put this way, I mean, there are different models, and models are simplified representations of the reality. Take animal model. You can mimic a human disease in the animal model, but that's it. That's how far the model will go. I mean, it's an animal. I feel that with the stem cells, we are still learning how to create better models. So in the beginning, we are used to do lots of neurons in monolayers. Now we can grow them with some organization in organoids. So all of that is still evolving. So we can also create organoids that are a little bit bigger because they have something that seems like a vascularization system. So you can improve upon the tools that we have. So we are always getting closer and closer to the reality, which is the human brain. So I feel that this is a promising model, new and exciting. So my lab has about 17 people now, 8 postdocs, some grad students, some master students, volunteers, undergrads, and technicians. So I feel that it is very lively atmosphere. Everybody is very supportive to each other. One good thing is that we all share the same tools. We're always using the same methodology, but asking different questions. So instead of a very competitive environment that you may find like in other medium-sized big labs, I feel that I was able to create something that was very collaborative because people can build upon each other and everybody grows together. So this is the kind of environment that we try to keep going here. And at UC San Diego, I'm also part of the Sanford Consortium for Regenerative Medicine. This is a new initiative from the SRM, which is the Stem Cell California institution. And together with a philanthropic donation from Denny Sanford, we were able to create this consortium, putting together lots of labs like mine that are using stem cells to learn about human diseases from the basic fundamental aspects to a more translational approach. So it's all in one. We are combining. We are constantly discussing how to take what we learn from the bench to the bedside, how to translate everything that we learn as fast as possible to the patients. So in a nutshell, this is an introduction to my lab.
Okay. Yeah. Can you talk about your mentors, role models in your career?
I think I had like really good mentors. My first Ph.D. advisor, Dr. Carlos Menke in Brazil. He was a very scientific, rigorous guy. Always wanted us to do the experiments that was perfect. Not afraid of repetition many times to convince ourselves that the data was really good. And once you have good data, you can build upon. So I think he was the first one to point to me that rather than rush in and have something just quick and dirty, but do something that is for sure. So you spend your time, but at the end you gain quality. So that was the first one, and I would say that my postdoc advisor, Rusty Gage, an amazing guy, full of energy, very creative, look forward, and I was able to also get inspired by him. And he in US, he was very helpful to point me to the right directions or to discuss alternatives. Coming from South of America, the US system of doing science is very different. When I start my postdoc, I barely speak English, so it was a lots to learn. And he was patient enough and kind enough to guide, to point, and to advise. So those two were really my major role models in science.
And in addition, I know you are a busy person with all the different projects going on, writing grants and all those things. So what are other hobbies do you have other than science?
No, here in San Diego, I mean, I learned how to surf.
Yeah, I know.
So, surfing is a good part of that. And in the outdoors, right? Really enjoying the outdoors. We have a son, he's 10 years old, and we like to take him out camping and explore hiking, just be in touch with nature. I think that this is good for my brain, who is so much into science, and sometimes I mean doing something different. It helps you to think more clearly, to give a break to your brain. So these are the major activities that we do when I'm not in the lab. That's where we're gonna find me.
Okay. Okay. And coming to the research, like what are the different projects? What are the different diseases you are looking at currently in the lab?
So the lab, I would say there are three major lines of research. They all converge into trying to understand the same thing, which is the big picture is the human social brain. What is so unique about the social brain? And why I'm interested in the social brain is because it's one of the characteristics that distinguish us from other primates. So the human brain really evolved to become a social entity. So from the beginning, from the day we are born, we are seeking social interactions. So for some reason, evolution thought that we are collaborative animals and we evolved to that, and probably that's why we are here. We can create art, we can create science, and other animals cannot do that. I think this is part because our social brain, the power of our social brain. So I try to understand that. And one way to understand something is to look for when something goes wrong, or the so-called mutants in nature. And a mutant for the social brain would be someone with autism, for example, that does not have that social drive. It is afraid, or sometimes do not know how to take advantage of the human social brain. So autism is a big part of my lab. We focus on different genes that can lead to autism, and on the other end of the spectrum, we have other types of mutants, genetic alterations that can makes you hypersocial. So this is, for example, what we see in Williams syndrome. People affected with Williams syndrome can actually become attracted to strangers. They like the new faces. They're so much more social than the normal. So that's a striking phenotype. And again, it's just like a mutation, a deletion in chromosome 7 that makes you hypersocial. So why is that? How those genes can actually change your behavior? So looking from a disease perspective, this is the portfolio of diseases that we choose to work on, mainly neurogenetic disorders that will affect your social brain. Then there is the other part of the lab that looks into that question from a more evolutionary perspective. So we try to understand how the human brain evolved to become a social brain. And this is, we do that by comparing the cells or the early stages of neurodevelopment from humans compared to other non-human primates, for example, the chimpanzee. So we have cellular models for those two species, and we compare, we use each other to contrast and see what are the differences. And the differences might appear at the genetic level, might appear at the molecular level, at the cellular level, or even at the network level. So we have different layers of complexity, and we just try to understand what's unique, really unique about the human brain. And finally, I have another line, a third line of research, and this is a follow-up from my postdoctoral work, where we figured out that there are regions, discrete regions in the genome called retroelement or retrotransposons. So these are quite ancient DNA regions, repetitive regions that are spread out through the chromosomes that have this remarkable ability to make copies of themselves and to insert back into the genome. So when they do that, we call them the jumping genes. When they do that, they expand the genome, and by expanding the genome, they provide a fertile soil for evolution to act, or even at the somatic level for some alterations in transcription, in splicing sites. So whenever there is a movement from one of those jumping genes, the cells become distinct. It is distinct at the genetic level because it now has a new de novo genetic scar into the genome, and it's different from the neighborhood cell. So it creates what we call a genetic mosaicism. And those insertions may actually affect how the cells behave. And we have some examples of that. A single insertion can make a cell form more synapses compared to another insertion. So we don't know yet if this is an important phenomenon in biology. It might be that it happens in the brain because neurons are postmitotic cells. They just don't care. They will not divide, so they are somehow protected. But it might be the other way. It might be that this is a very conserved mechanism to generate genetic diversity in your brain. And this diversity might actually be important on how you think about everything. So it might be the factor that makes us individually unique. So we work with those two hypotheses, or three, let's put this way. One is a positive hypothesis, so those things are improving the fitness of the individual or the species. There's a neutral hypothesis, they're just there, we don't care, or a detrimental hypothesis. Whenever it happens, it's actually bad for you. And on support of this third hypothesis, we have been having some correlational studies between mental disorders, autism, and schizophrenia mainly, with a high level of retrotransposition. So it seems like if you have too much, it's probably bad for you. But again, I mean, this is early stages of a very unexplored field, and we don't know where it's going to take us. So it's, I would say this is a more exploratory side of the lab.
Okay. And what are you going to present today?
I'm going to talk like an overview of different projects in the lab using iPSC with mutations in some genes that cause that affects the social brain, mainly in autism.
Can you please go ahead and do the presentation? Thank you.
Yeah. Let's see if we can share. All right. So I believe you are seeing the first slide now.
Yep.
All right.
Go ahead.
Okay. So as I mentioned, major focus in my lab is to explore autism. And autism is not only interesting from the aspect of the human social brain, but it is also a condition that affects many people. Actually, 1 in 54 boys will be born with autism. So that's a quite high number. And those numbers have been changing lately. We mainly have data from US and England, but it seems like quite conserved across the world. We think that the increase in the numbers are probably, or in frequency, are probably due to a better diagnostic. People know more about autism. Doctors are more familiar. We are talking more about the condition than we did in the past years. We don't know much about autism, but we know that in some cases there is a strong genetic component, and in other cases it seems like it's more driven or caused by environmental causes. And we don't fully understand how those two interact. I mean, we know that probably they do interact with each other, but it's unclear yet how this happens. And because we have so many people with autism, this is quite a burden for the system. So economically, it's quite heavy on the state, on the government, but also for the families. This is too much. So this is what we expect as a cost during the lifetime of someone with autism. 3.2 million, and these will go into special schools, special educations, therapies, and all the service that they need. One has to remember that people with autism experience the world in a different way. So even simple task as going to the dentist is not easy for people with autism. So they may require a special dentist that knows how to deal with the condition, that has a special settings to treat people with autism. So these all increase the cost of living with someone with autism. More importantly to us, the fact that there is no cure, there is no such a magic pill that one can take and revert autism. So this is quite frustrating. Most of the medicines that we have out there, these are for to treat the symptoms of autism. And one example is the fact that 20–30% of the people with autism might develop seizures at one point in life. And so we can take drugs to control the seizures. Same thing is for GI irritations, and you might take like some drugs to control that. But really, for the core of autism, there is no drug that will make you more social, for example, or we speed up your learning ability, or improve your language. So these are core deficits of autism that we have no medications, no cure. And as I mentioned in the past, the model that we use is induced pluripotent stem cells, so we can reprogram somatic cells from individuals with the condition, push them back to these early embryonic stage, or iPSCs, and from the iPSCs we can make brain cells. And then we compare these brain cells derived from autistic individuals to a group of controls, non-affected individuals, and we look for differences. And those differences might be at the molecular level, might be at the cellular level, the circuitry level, but nonetheless, if you find one of those differences, you might use that as a readout in a drug screening platform, for example. That will help you to speed up drug discovery process. So we're very interested on this translational aspect. So when I started my lab, I didn't know exactly how to start, but I thought that was important to start with well genetically defined conditions under the umbrella of autism. And Rett syndrome is one of them. The clinical phenotypes are more homogeneous. It's still variable, but way more homogeneous than overall autism. And we know it's caused by mutations in a gene called MECP2. So we recruit individuals carrying different MECP2 mutations, both boys and girls, and we use skin cells or fibroblasts as a source to reprogram these cells, and we start making neurons from Rett syndrome patients. And the interesting part was, in the beginning, it wasn't clear what to look for. Nobody has done that before. So I have to take you back to like 2008, 2009. And there was one clue, one paper describing morphometric alterations done by Golgi staining in a brain of a boy with Rett syndrome. So you can clearly see in the bottom here, you have different cortical layers staining by Golgi from a control case, and on top from the MECP2 knockout or mutant individual. And it's pretty clear that those neurons are less arborized, they are smaller, so visually they look like immature compared to controls. So we thought that morphology might be a best way to do it. So this is the protocol that we have. We keep improving the protocol, but we start with a well-defined condition media, and we grow the iPSCs. From the iPSCs, we make embryo bodies, and from the embryo bodies, we make neural rosettes. And if you dissociate these neural rosettes, they will become neuroprogenitor cells. And on the bottom, there are different markers that will tell you that the steps that you have are the right ones, and you can be confident that if you just withdraw the growth factors from the population of neuroprogenitor cells, they will spontaneously differentiate in what we call a mixed neural culture, or a heterogeneous culture, because we have both excitatory neurons, inhibitory neurons, and several astrocytes in there. Depending on the assay, you can use FACS or MAX, magnetic cell sorting, to isolate specific subtype of neurons or specific subtypes of glia cells. And this is how the cells look like if you plate the neuroprogenitor cells in the absence of growth factors. On the top, you have a cluster of cells, and they would migrate out. They will follow the process of other cells that have previously migrated. And as they approach the border, these cells will stop, differentiate, and start making connections with the neighbor cells. And we characterize these cells using different markers, different technologies. More recently, we have been focusing on single cell analysis because we want to know what kind of neurons we have in our dishes. So this is one example of a Fluidigm machine where we can capture single cells using this Fluidigm platform. And in each cell, we can analyze the expression of different genes. And from this data, you can explore if the control group are similar to the rat group. So in the bar graphics here, what we have is the controls in blue and the Rett syndrome in red. And just from the beginning, you can tell that layer 5 and 6 from Rett syndrome are not as well represented as we see in controls. And already see that the number of glutamatergic neurons or cholinergic neurons in the Rett syndrome population is also not as represented as the controls. So the differentiation is not identical between these groups. So there are differences in the type of neurons that we have. On the other panel here, we just have immunostaining with different cortical layer markers. So we can be confident that those neurons are indeed glutamatergic pyramidal cortical neurons. So these are 80% of the neurons are this type of neurons that we can get from the iPSCs. In this next slide, there's just a different analysis where we can use the 10x. It's a more powerful technology. So virtually you can do RNA-seq in single cells, and you have those maps that display all the populations that we have. And similar to the Fluidigm machine, we can see that the population of glutamatergic neurons here in blue takes a large fraction of the population. We do have a small population of GABAergic neurons. There is also some progenitor cells going on. And in red, you can start to see some glia cells. Most of it are astrocytes. And in this slide, we have the first phenotypic data from Rett syndrome, just showing that if you measure the soma of these cells, they are not as big as the control. So the soma size is slightly smaller compared to the controls. Another dramatic phenotype we see in those neurons is the absence of neuronal spines. In the images here, you can see that the white type or the control neurons, they show lots of spines. In the Rett syndrome-derived neurons, we don't see it. All these phenotypes you can validate using shRNAs against MCP2 in the control or the white type neurons to mimic the phenotype. So we are confident that this is caused by the mutations in the MCP2 gene. But the most dramatic phenotype, it is the number of synapses that we see in those neurons. And I will just play a video here of a panoramic perspective of the synaptogenesis that we see in these cultures. In the left, you have your control cells. In the right, your Rett syndrome-derived neurons. And it's pretty clear that although the number of neurons are similar in red, the number of synaptic punta in red here are reduced in Rett syndrome compared to controls. And it's a pretty dramatic reduction. So they don't make as many synapses as we see in controls. And this is the quantification. So it's a little bit lower than 50%. But the good part is that this is not something permanent or fixed. If you expose these Rett syndrome neurons to specific drugs such as IGF-1, insulin growth factor 1, you can actually revert that. So the Rett syndrome neurons would behave similar as the control group. So it suggests to us that these conditions can be reverted eventually if we know how to do it, if we have drugs that can actually penetrate into the brain and induce the activity of those synapses. So everything that we did with the immunostainings can be recapitulated using electrophysiology. Here is our collaborator, Gong Chen, who did that in a blind fashion. So you can patch clump control cells or Rett syndrome cells, And virtually after doing lots of recordings, he realized that the Rett syndrome cells have a decreased frequency of spontaneous postsynaptic current, similar to what one would predict with a lower number of excitatory synapses. So it's always nice when you have different complementary technologies that point you in the same direction. So this is possible with neurons in 2D cultures, but as I mentioned, we can use this technology to grow cells in 3 dimensions. And this is what we call cerebral organoids, where we have some kind of organization. There are ventricular zones that might keep stem cells or progenitor cells, and those cells will migrate out and form the cortical plate. Once they reach the cortical plate, they can also migrate both radial and tangential in a tangential pattern, and we'll start to see some kind of organization. So the cells look more organized. As these organoids mature, you can see a better definition between the cortical layers and the electrical activity as well. We use several markers, again, similar to what we did in 2D, to visualize these different cortical layers. And at the functional level, we did several experiments. This is just a video of a voltage gated dye. So you can see that neurons are pretty active, so they are spontaneously firing inside these organoids. So this was done with a confocal microscopy, two photon microscopy. We can do the same thing with a calcium dye. And again, I mean, you can see calcium transients coming from these neurons, and you can trace them, and you can basically confirm that they are active. But to me, the really cool functional analysis is when you plate these cerebral organoids in a multi-electrode dish, and this is something that was developed by a former postdoc in the lab, Cassiano, and he basically places some of those mini brains on these multi-electrode wells. In this case, we have a 8x8 or 64 electrodes, and these electrodes will capture the electric activity and will generate an activity map similar to this one that I'm playing here. So the more the red, the higher the frequency and the intensity of the bursts. The machine will actually spit what we call a raster plot. Here on top, we have examples of a raster plot. Each channel is one line, and each trace is a spike event or a neuron that's firing. And if you have too much of those spike events in a very short of time, we call that a burst. And if you have bursts that aligns in several channels, we call that a synchrony event. And over time, we see that these cells tend to reach to a more synchronized event. So this is just over time what happens. At 8 weeks, we start to see some activity. 10 weeks, the level of activity becomes more frequent. And at 12 weeks, not only the activity is increased, you have way more activity, but the number of synchronization events start to appear, start to become clear. So this is what happens in a normal situation. Now, if you compare that with Rett syndrome organoids, we don't see as many synchronized events as we see with the control group. So this is, again, another indication that Rett syndrome neurons will not develop as the controls and will not reach out to this level of maturation. An interesting approach that we also use is to take advantage of CRISPRs, or genome editing enzymes, to create mutations, or in this case, to rescue the MCP2 mutation. And by doing that, we were able to fully recover the electrical activity. Those neurons can now mature similar to controls. This is what the bar graph here suggests. If you see this small r follow the RTT in the gray bar, this is our genome edit rescue cell line. So no longer carry the mutation, and the number of synchronicity increased to the control level. So this is pretty cool. So we are using these kind of readouts in a drug screening platform. For example, we team up with the NCAT at the NIH, where they have these robot arms that will do the screening of thousands and thousands of drugs for us. And in the bar graphs here, I have just some examples of drugs that might actually increase the number of synaptogenesis. The last bar is IGF-1, so you can see that there are drugs that are as good as, if not better than IGF-1. So again, I'm very optimistic about this possibility of finding new drugs and apply them to the patient. And so some of the drugs can actually rescue the networks. So this is using the multi-electrode array. But another thing that we can do with those cells is to dissect the contribution of individual cell lines. And you have, for example, glia cells. I mentioned that we can make astrocytes from these neuroprogenitor cells. We use a protocol that virtually gives you a very homogeneous or even pure population of glia cells after sorting. And it's a quite fast protocol. This was developed again by Cassiano together with Bia, two postdocs in the lab. The protocol is quite simple. Instead of creating neurospheres, you change the medium to a medium that favors the appearance of astrocytes, and they would migrate out from these astrospheres, and at the end you have this population of nice astrocytes. So we use now astrocytes to compare gene expression between controls and rats, and we see different things. One example is genes that are highly expressed in astrocytes, such as GFAP, aquaporin, S100 beta, or vimentin. Those genes are always upregulated in Rett syndrome compared to controls. An indication that the Rett syndrome astrocytes are kind of more reactive compared to controls. Again, we don't fully understand how this happens. Other phenotypes that we could see is, for example, on the distribution of calcium waves after an injury. The red circle here suggests that it points to the cell line that we touched with a pipette, and then you can follow the calcium waves spreading out in a normal situation. In Rett syndrome astrocytes, they are unable to spread the calcium after the injury. So again, there is something wrong with those astrocytes that we don't fully understand. But those are phenotypes that guide us to do a more careful analysis, such as a co-culture experiments, where we're going to plate neurons on top of a monolayer of astrocytes and see how the astrocytes can instruct the neurons to behave or to differentiate. And in this next slide here, I have an example of what these astrocytes can do if you just mix different genotypes. For example, Y-type astrocytes, Y-type neurons. The neurons develop quite well. I'm just showing here the neurons in green. If you plate the same control neurons, but now in a bed of Rett syndrome-derived astrocytes, they fail to thrive. They have this bipolar looking, not many arborizations going on. The same is true if you have Rett syndrome neurons and Rett syndrome astrocytes. Nothing really happens. They're immature. But to me, the most dramatic data is when we plate those Rett syndrome neurons. Remember, they carry the genetic alteration, and you place them in a bed of normal astrocytes. You fully rescue the morphology. They now look way better. They are indistinguishable compared to controls. We can actually quantify all these different parameters, number of dendrites, neuronal length, segments, and even number of synapses, and they do look like normal neurons. So again, it's a mutant neuron, but in the right environment will behave like a normal neuron. So this is very encouraging for us, and we have been looking for what could be the toxic factor. Oh, I apologize for that. the toxic factor coming from the astrocytes. Again, we use gene expression to learn that Rett syndrome astrocytes, they have a misregulated signature of cytokines. And among the most upregulated cytokines, we have IL-6. So the bar graphics here points to a high level of IL-6 in Rett syndrome mutants or in Rett syndrome artificially generated knockout. So by removing MECP2 from the cell, you increase the levels of IL-6. This is something that we also see in postmortem brain tissues as well. So IL-6 is a pro-inflammatory signature. So we thought that what happens if we just expose normal neurons to the same levels of IL-6 that Rett syndrome astrocytes can generate? And the truth is that IL-6 at these very high levels is very detrimental. And this is just one example of morphology. We see a decrease in the total neuronal length. The number of synapses is decreased as well. And the overall spikes coming from the multi-electrode array is also decreased in the presence of IL-6. So IL-6 seems like one of these key molecules, a cytokine that might actually contribute to Rett syndrome. So we are further exploring ways of taking advantage of that information and use, for example, IL-6 blockers or neutralized antibodies to see if we can ameliorate or improve the condition of Rett syndrome patients. I'll also mention that we are not only studying Rett syndrome. Again, we start with Rett syndrome because it was an easy entry to the question, to the biology, because of the well-defined genetic alteration. But we have been collecting cells from autistic kids for the past 10 years. And we do that using social media. We create the Tooth Fairy Kit collection, where we connect with the families using Facebook, for example, and we send a kit with instructions for the families to send out their milk tooth to the lab. And from this milk tooth, we are able to isolate the dental pulp cells and create iPSCs from these materials. So there is no need for the families to come to the lab for a skin biopsy, for example. So that removes lots of stress from the families to come to the lab. And by having a large collection of these cells, we can now model different types of autism. And one condition that we have been looking are the ones with macrocephalic brains. So there's a population of autistic kids who have big brains. The volume of their brains are bigger compared to controls, compared to the normal curve. And it's only a fraction of them, but it's a large fraction. It's about 20% of them have these big brains. And if you just do a correlation between the number of Ki67 cells, these are proliferative neuroprogenitor cells, there's a nice correlation with brain volume. So the autistic-derived neuroprogenitor cells, they actually proliferate faster or more compared to controls, and that might explain why they have these enlarged brains. And these enlarged brains, one would expect them to form more synapses, but we see the opposite. We see that more neurons does not necessarily mean more synapses. They actually have lower level of excitatory synapses. And this is reflected on the overall network formation. So the number of spikes and the synchronization is way reduced in autism compared to controls. So the bar graphic here is on the right. You can see just by the scale that is so much different. So basically, these autistic-derived networks, they don't make lots of connections. And we have been testing IGF, similar to what we did with Rett syndrome, in idiopathic autism. And what we get is something quite interesting. It is not all the cells responded in the same way. So what we have in those graphics here, it is the number of spikes or the electrical activity from the multi-electrode array in non-treated. This is the black curve, or treated with IGF-1, and the day of treatment is indicated here by this line here. So all of those are autistic-derived networks, and you can see that the top ones, for example, after adding IGF-1 to the culture, the number of spikes go up. They responded really well, really fast to IGF-1. In the bottom ones, you have like 3 networks that do not respond as well to IGF-1. They actually took a little bit of a lag of time to respond to the network when they responded. So we think that this is a very powerful tool to select the cohort that we have to move on into clinical trials. For example, if I'm going to do like a clinical trial for IGF-1, I'll probably only recruit or expect only the top 4 to respond to IGF-1 compared to the whole population, because it's so heterogeneous. So we are trying to understand why this happens at the genetic level as well. But nonetheless, these mini brains or organoids in a dish can actually point you to who is going to respond to a specific drug and who are not responders. So it might save lots of money in future clinical trials. So I'll actually finish here so I have time to discuss a little bit by thanking the members of my lab. I pointed to some of them on specific projects here, but also for the families and subjects who are also very connected to my lab, which is something that I really appreciate, and always helping in not only with materials, information, but also giving insights into the research. So I'm very grateful to have this social network of families integrated, plugged in with my lab, so people appreciate that as well. So I mean, this is the slide that I have. I'll finish here. I don't know if you want to discuss some of the data or have any questions for me.
Yeah, certainly. You mentioned that at the end, like there is that discrepancy in like not everybody responds to the same thing, but they have similar phenotypes in terms of neurons which are not responding very well and all. So do you think it is genetic, at the genetic level, the difference comes in?
I think so. We have at least some indication that the genes that are mutated in those patients that are responders versus non-responders, they are slightly different, and that might affect the metabolic pathways that IGF is using to increase neuronal maturation or to increase synaptogenesis. But the number, the number of patients we have right now is quite on the lower side to make any conclusions about that. I mean, one would have to do that with 100 patients or even more to make sure that this is the direct link. The technology is so expensive, so it's so expensive to reprogram cells from patients, to do all these analyses, that we cannot afford to do it with large cohorts of patients. I think as we reduce our costs per patient, we can do more and more. But right now, this is one of the limitations of the technology.
And these IGF and all, are these like, do they enter the brain, and how does it go? Like, are you looking at some of the molecules which will enter the blood-brain barrier?
Yeah, that's a great question. IGF, as we tested now, it's a very, it's a big molecule. And we have the full-length IGF-1 in a dish because we can just expose them directly to neurons. Thinking clinically, one will have to infuse IGF-1, and very few would actually get to the brain. So there are companies looking for small molecules or peptides that can mimic what IGF is doing and penetrates much better in the brain. So there are actually clinical trials going on, both with the full-length molecule as well as with small peptides that that might work better. So these are all in clinical trials now.
And again, going back to the IGF, like IGF might be involved in various other things, so metabolic disorders and all those things. Have you looked at any connection between these and the metabolic disorders?
In some cases, we do have a clear evidence that the individual has a metabolic disorder as well on top of autism, and which may suggest that IGF might really work on those cases, work much better. There are some indications in the literature that some of the patients that we think has autism, they do have a metabolic disorder. And I think those cases would be easier to treat. But to do that, unfortunately, you need genome sequencing. And this is something that is not on the clinics yet. I mean, doctors are not prescribing routinely genome sequencing to all autistic patients. So I think in the future, this should be recommended. Right now, probably it's a cost issue, not and also, I mean, knowledge. Not many doctors are prescribing that, but I can tell that in the near future, we're going to learn more and more about the variants, the genetic variants that will help us to personalize the treatment to a specific individual. And IGF might be one of those treatments that will work really well in some of those patients with clear genetic metabolic disorders.
So have you looked into any other than IGF? Like you talk about various like the problems with in terms of spines, not having spines, synapse formation problems. Is there any cytoskeletal related stuff involved in that?
Yeah. So both at the causal level, I mean, we can see that some of those genes, when mutated, they are involved in cytoskeletal organization, and that might explain why they do not make as many synapses as one would expect. But we have been exploring other drugs as well. So we have several modulators of neurotransmitters that can actually act indirectly into these networks and correct the network to the control level. So these are, again, drugs that can modulate specific neurotransmitters that we identify as being low or high in the disease state compared to controls. Another possibility, as I mentioned, I mean, since the Rett syndrome brains have these high levels of IL-6, one can neutralize IL-6 in the brain and see if that helps. So we have those reagents now. There is actually one drug that has been using for arthritis to inhibit IL-6, and we're gonna try to repurpose that for autism, Rett syndrome autism. So we have to do all the preclinical stages before we move into the clinical phase, clinical trials. But this is certainly something that was unexpected to us. I mean, we knew that in autism there is an inflammatory component, but we never thought that would be causal. We thought that this was a consequence of the disease process. But now I'm changing my mind. Actually, the data is showing us that no, it's actually causal. So if you block IL-6 or reduce IL-6, you may ameliorate the condition.
Okay. I think we are almost out of time. I really thank you for taking time to do this, and certainly it would benefit a lot of viewers. And I really appreciate your time, and good luck with your future work, and thanks again.
Thank you. I hope I convey a little bit of the science that we do. I hope it was clear. And if not, I mean, feel free to contact me. If you guys have more questions, I'll be happy to answer them. And congrats to you for putting this together. I feel that again, we need more science speaking in open platforms like this one.
Thanks again. Thank you.
Thank you. Bye, guys.